Gravity dictates how your cells physically remember orientation. The structures inside us respond not just to chemistry, but to subtle pulls in space itself. Imagine a river flowing always downhill. That simple constant directs the delicate scaffolding within every living cell. That internal framework, called the cytoskeleton, is intensely tuned to these external directions. It measures them without needing eyes or sensors. It acts like an ancient compass built right into its core components.
But how does that internal compass translate a simple downward pull into specific cellular direction? It turns out the cell is far more than just passively feeling weight. Its fundamental blueprint aligns with universal rhythms. Imagine every single protein inside us subtly seeking an invisible magnetic north, even when no magnet is present. This inherent alignment toward external physical vectors dictates where energy flows and what signals fire within these systems, influencing those critical internal pathways we observed earlier. It suggests our own structure possesses an innate tendency to match the deep cadence of existence itself.
What if that alignment isn't just passive obedience? Imagine every cell actively participating, translating those slow spatial cues into fast internal chatter, like tuning a delicate instrument against a steady hum. We are moving beyond simple sensing now. The research suggests how variations in baseline physical direction can tune the very electrical coherence inside our complex systems. Think of it as micro-adjustments to the cosmic radio frequency, affecting neuronal whispers. This mechanism we saw earlier, that directional bias toward universal rhythms, starts correlating with measurable shifts in bioelectrical states across networks, which means something nobody expected for how intrinsic cellular tension interfaces with larger wave patterns. Next up, we find out what happens when those tensions get stressed even further. But what happens when that established structural bias gets pulled by something else? When internal mechanical tension meets an external invisible force gradient beyond simple directionality. Think about tuning a grandfather clock in a building whose foundation is subtly vibrating because of traffic miles away. That's the kind of subtle environmental crosstalk we are now seeing influence molecular activity on a quantum scale within cells. Researchers like Dr. Ito have suggested these low-frequency field variations can actually warp protein folding kinetics itself, far outside conventional chemical models. Does this imply our internal state isn't just listening to gravity, but perhaps feeling something much deeper? And who is right if the traditional rheology theories still insist everything must be about bulk flow? You absolutely need to hear how that connection scales up next. We saw earlier that structural bias connects to electrical coherence, but what happens when those intrinsic tensions meet a specific external push? Imagine trying to balance an impossibly delicate gyroscope on its edge while standing in a hall where someone keeps humming low frequencies just below hearing range. That subtle environmental input isn't noise, it's information being registered at the quantum level within cellular architecture itself. This means that those slow spatial cues we discussed earlier aren't merely measured by our cells like temperature, they are actively modulating protein interaction kinetics through what theorists call etheric grounding. The cell becomes a subtle resonator to every pull in existence, and that resonance dictates its internal state towards something far beyond simple mechanics. What is the cellular equivalent of intentional alignment? That realization leads us directly into how life calibrates itself against universal rhythms next. But what if that alignment towards universal rhythms isn't just following a path? What if our cellular architecture is actively seeking an innate directional bias? Imagine every internal scaffold has an innate preference, like specialized riverbeds naturally favoring specific geological flows, even when submerged. The research shows this tendency aligns with how cells manage their own deep orientation cues through processes we call endogenous polarization alignment. This suggests that the physical environment doesn't just dictate what happens, it subtly biases the entire spectrum of cellular potential itself toward a preferred cosmic setting. That innate bias, which seems almost like intention within biology, changes everything about how we view living structure next. The whole story points toward one fundamental thing. That built-in cellular tilt, that preference seen earlier, isn't random compliance. there is an active mechanism translating those subtle external tilts into internal life chemistry. Think of our cells not just as passive receivers, like a fishing net catching drift debris, but as finely tuned tuning forks resonating to the ambient hum of existence itself. The real breakthrough lies in recognizing that this mechanical registration feeds directly into how energy flows within us, how we maintain our inner coherence across vast distances inside the brain. This connection between physical grounding and internal electric rhythm is where modern theory finds its edge, pointing towards endogenous modeling frameworks for what happens when those boundaries shift slightly. That insight gives you a way to approach your own baseline orientation with newfound awareness. The full research archive is at jmthomasofficial.com.
The verdict is clear then, that what defines our internal state isn't just a static reaction, but an active calibration toward inherent directional preference. Think of it like specialized magnetic silt settling only in certain orientations within deep oceanic currents. Life seems biased toward these specific energetic settings, even when everything appears uniform around us. This innate alignment suggests we might be tapping into endogenous modeling frameworks that connect subtle physical boundaries right to our core bioelectrical coherence. It moves the study from simple measurement into genuine understanding of intrinsic cellular potentiality itself. The full research archive is at jmthomasofficial.com.